All eukaryotic cells consist of the same DNA, but why don't they all perform the same functions? This is due to eukaryotic gene expression, and will be my topic for today. Gene expression is the activation of a gene that leads to transcription of mRNA and overall results in the production of a protein. This process allows certain genes to be expressed depending on the need for the individual. There are many factors that can affect gene expression that depend on the internal and external environment of the individual to determine what is needed to be expressed. Typically, in eukaryotes, gene expression is regulated at the transcriptional level. The initiation of transcription is regulated by transcription factors and transcriptional regulator proteins. Many transcription factors can control multiple genes, but these genes are commonly expressed at different levels and at different points in time. To initiate transcription, the transcription factors bind to a specific DNA sequence, or the promoter sequence. The relationship between the promoter and the transcription factors is crucial to the diversification of gene expression profiling. At the start of transcription, a basal transcription apparatus binds near a specific promoter sequence called the Tata box. Transcriptional activators and coactivators bind to the DNA sequence to assist in stabilizing the apparatus at the core promoter. The transcriptional activators initiate the transcription. In addition to transcriptional activators, there are also transcriptional regulator proteins. These consist of transcriptional repressors, enhancers, and insulators. Transcriptional repressors bind to silencers, which prevent the genes from being expressed as proteins. Enhancers are a DNA sequence that stimulate transcription upstream from the promoter sequence, and insulators are DNA sequences that block the effects of the enhancers. Insulators block the effects when they bind between the enhancer and the promoter. On the left side of the slide, there are three pictures. The top picture is an example of a transcriptional repressor binding. The middle picture is an example of an enhancer binding. And the bottom picture is an example of an insulator binding to the strand. These different control mechanisms are used as a way to conserve energy. It would not be energetically efficient for a cell to express every gene it contains at all times. With the assistance of these transcriptional regulator proteins, a gene is able to turn on the necessary genes only when they're needed. Introns are also used as a way to control gene expression. Introns are sections of DNA or RNA that have been spliced out prior to translation. Introns are non-coding sections of DNA or RNA and interrupt the sequence of genes. Introns have been found to increase the efficiency of transcription in many genes throughout nearly all organisms. Introns can act as a storage unit for transcriptional regulator proteins such as enhancers and repressors. They can also control the DNA accessibility through a process called nucleosome positioning, which tells where the nucleosomes are with respect to the DNA sequence. Introns also contain splicing signals, which can enhance RNA polymerase II initiation, stimulating transcription. Introns are a good example of regulating gene expression without acting as a binding site for transcription factors. All of these transcriptional regulator proteins play an important role in gene expression. They assist in controlling which proteins are present in the cell based on the function of that individual cell. For example, it would not be useful to have a protein that is present in the liver that breaks down alcohol be present in a neuronal cell. The protein that breaks down alcohol in liver cells is known as alcohol dehydrogenase, or ADH. ADH is broken down into three classes: Class 1, Class 2, and Class 3. The liver has the most abundant source of Class 1 and Class 2 ADH, but there has also been activity of this enzyme in the lung, kidney, and gastrointestinal tract. Class 3 can be found in all human tissue types. ADH is regulated by the cell's external environment and is the response to an increase or decrease in the blood volume. This variation demonstrates that each cell contains specific genes that are expressed based on the function of that particular cell in order to make the cells as energy efficient as possible.